Welcome back, guys. Today we're going to be starting our two-day foray into cell structure and function. So this is kind of your more conventional talk about organelles. So hopefully a lot of this is review for you guys from previous classes. We're going to be splitting it into 2 days just because, as you've probably figured out by now, nothing in AP is ever simple. We're going to be going more in depth into the role of each organelle, and you will be expected to have a deeper understanding than you typically would. So while you're taking notes, I would suggest maybe doing 2-column notes or doing their notes in outline form, just so that you can get the name of the organelle and what it does. This is going to be important because tomorrow I'm going to be discussing the project that y'all will be doing with this. It's not going to be the same project that Biology 1 is doing, so don't even start thinking that kind of way. But yeah, so be on the lookout for that. And also you'll be doing a case study today. So it should be pretty self-explanatory. You read the information and you answer it, but I can show you guys at the end of the video. Anyway, so cell structure and function. Okay, so cells in general, they're the basic structural and functional unit of every organism. So what this means is the smallest that I can get and still carry on the processes of life is a cell. Any smaller than that, and it doesn't really do that. So all cells, this sounds like a great test question, are bound by a plasma membrane. They contain cytosol. So some books are going to refer to cytoplasm as cytosol. This is just the jelly goo inside the cell. Okay, they contain chromosomes, so that's, DNA. They contain ribosomes, which everybody's got to make proteins. Oh, okay, so these 4 things, this is yes, all cells do this. So just make sure you're able to recognize that and answer that. Okay, so there's 2 main types of cells, prokaryotes and eukaryotes. The biggest thing we're looking for, does it have a nucleus or does it not have a nucleus? So prokaryotes, this is the domains bacteria and archaea. So this is information right here that we don't really cover in the AP curriculum, but domains, like even before kingdom, like King Philip came over for good soup, kingdom, phylum, class, order, all that, up above it, we have 3 domains, which is bacteria, archaea, and eukarya, all right? So there's 2 whole domains of prokaryotes because there's just that many bacteria out there. So their DNA is in the nucleoid region. They don't have an actual nucleus. They have a big, like, spaghetti mess of DNA, or they have it in short little circular segments called plasmids. So they're generally smaller in size. So if you Google search pictures of it, I don't think there's one in this presentation, but when bacteria attack a cell, like a body cell, these bacteria look like little grains of rice on top of a gigantic booger. Like, they're so much smaller, it's ridiculous. So eukaryotes. So these are like kingdom Protista, fungi, Animalia, and Plantae. So protists, I don't know if we're going to get into that early, but protists, they have a nucleus, but they're single cellular. That might actually be worth a Google search if you get bored, especially Chrysophyta. They're super pretty. I'll try to, if you're listening to this and you want some more information on that, I have some really cool videos I can share with you. So DNA is going to be kept in a nucleus in a eukaryote. They have membrane-bound organelles. So what this means is, instead of just throwing it all into this itty-bitty little cell, eukaryotes can subdivide their stuff with membranes, and we don't see that in prokaryotes. All right, so this is a microscopic image. They have stained different regions of the cell, but this is an electron microscopic image. So we can see in the bacterium, the blue part is its nucleoids. They've probably stained the DNA. You don't really see a hard edge to it. It's just kind of there. There's not a whole lot of sacs at anything. It's just all thrown in there. In a eukaryotic animal cell, we can see there's little bubbles in there. So all of those are different organelles. In this particular electron microscope image, it's a little bit hard to tell what the individual organelles are. So whenever you have to label one, obviously we won't use something like this, but we do know the big blue part, that's where they've stained the DNA, just like in the bacterial cell. So you can just side by side see a difference. And then we see the cell membrane is that thick kind of outer layer there. Okay, so now to the organelles. So organelles are membrane-bound structures in eukaryotes. We technically throw ribosomes in there even though they aren't membrane-bound, but it is what it is. So there are endomembrane organelles. I'm just going to click this so it brings up the list. And then there's energy organelles. So we are only going to cover endomembrane organelles today. We are going to leave energy organelles and then like the mop-up for tomorrow, because believe me, this is plenty. So the idea of compartmentalization, this is a big nasty word that basically says that cells are divided up into different rooms, I guess. But it allows for different reactions to occur in different locations. This can be important because, you know, when we we do a chemical reaction, sometimes those chemical reactions can interfere with each other. So if I need to do both, but I don't want them to mix, I need to make sure that I make like a little pouch or a little room over here where this one happens, and then one over here where something else happens. And so sometimes we even have compartmentalization within the organelle. So it can have like a mitochondria. It's doing one thing in the folds, but then in the liquid area outside the folds, it's doing something else. So this also increases surface area. So surface area is super important in a chemical reaction. So like if you want your ice to melt faster, or if you want something to dissolve quicker, you crunch it up because that increases surface area, right? So if we want to increase the rate of chemical reactions, we have to increase surface area. All right, so there's some organelles that are unique to plant cells and some that are unique to animal cells. Oh, hold on. Okay, you are going to need to know this. So chloroplasts obviously are only in plant cells, with a big asterisk that when we get to microorganisms, sometimes they do weird stuff, okay? But for our purposes, okay, plant cells have chloroplasts. They have a large central vacuole. That is referring to like the big water balloon that you see in the plant cell down there. They have a cell wall, and they have plasmodesmata, which basically means they're little portholes in the cell wall, because we want to be able to get things in or out. So animal cells, they have lysosomes. Plants don't have those. They have centrosomes and they have flagella. So flagella, they're like the whip-like tails. All right, so our endomembrane organelles. So we have a nucleus. It stores chromosomes or genetic information. It's enclosed by something called the nuclear envelope. So a nucleus has a double membrane with pores in it, almost like it's its own cell. So those pores regulate entry and exit of materials from the cell. So like when I make an RNA message, I have to be able to get that out of the nucleus somehow. It can't be trapped in there. So the arrow's pointing to a nuclear pore. So it also contains something called a nucleolus. So when you see a cell diagram and there's like that dark patch in the nucleus, that's the nucleolus. It's a dense region where ribosomal RNA is made. So ribosomes get made there. So ribosomal RNA is combined with proteins to form the large and small subunits of ribosomes. So the subunits have to exit from the cellular pores because everything that goes in or comes out has to go through those holes. Okay, so ribosomes, they translate the messages found in messenger RNA into the primary structure of polypeptides. So simple version, ribosomes put proteins together, okay? They take each amino acid and put it together in sequence based on an mRNA message. We're gonna talk more about that process in another unit. So here's a scanning electron microscope image of this. This is what a nucleus looks like under like intense magnification. So we can see the outer membrane, the inner membrane, and the nuclear pores here. So ribosomes, they're composed of ribosomal RNA and proteins. So as we said, they synthesize protein. That's what they do. We have them in the cytosol or cytoplasm, basically the jelly area just floating around in the cell. And they're also bound to the endoplasmic reticulum or nuclear envelope. So that's why the rough ER is rough, is because it's coated with these little ribosomes. It looks like it's got bumps on it. So these proteins, so the proteins get secreted or used in the cell. That just means that when I'm making proteins, either they stay in the cell or I can release them because they're antibodies or a message or something. So if I want to get something out, we can send the proteins out through vesicles. A vesicle is just like a messenger pouch, like you just put it in a pouch and you send it out. It's a little membrane sac. Okay, endoplasmic reticulum, which is a long, nasty word. So it's just a network of membranous sacs and tubes. So on these pictures, you see the little foldy tubes around the nucleus. That's our endoplasmic reticulum. So it synthesizes membranes. So when I want to make phospholipids to make cell membranes or to make membrane sacs or whatever, those get made in the endoplasmic reticulum. It also compartmentalizes the cell to keep proteins formed in the rough ER separate from those of the free ribosomes. So let's say I need to make a ton of insulin to help sugar uptake, right? I may not want to keep all my insulin in that cell. That would be really bad. It wouldn't be able to use any sugar itself. So we want to make sure we keep those separate and we can get them out of the cell and where they belong. So there's two types of ER, as we said. There's rough and smooth. The rough one is focused with making proteins like insulin. And the smooth ER, that's where we're making lipids. So like the phospholipids for membranes get made there. Oh, hold on, there we go. All right, so the rough ER has ribosomes. I feel like this is repeating itself, so I'm just gonna click through it. But here's like a diagram. See how it's coded with the ribosomes. And the smooth ER has none. It also, another role of it is it detoxifies the cell. So it can help break down chemicals, basically, in the smooth ER. All right, so here is a cross-section of the endoplasmic reticulum. We see there's a bunch of free-floating ribosomes, but there's also some that are attached to this membrane here in the picture. So the Golgi complex or Golgi bodies, the terms are interchangeable. There was an Italian scientist whose last name was Golgi, and he figured out these exist, so now we've got that. So it contains flattened sacs called cisternae. So a cistern is like a storage place for rainwater. So these flattened sacs are cisternae, okay? So it separate, okay, hold on. So the cisternae are not actually connected. These sacs aren't connected. They're just stacked on top of each other, and there's something called directionality here. So there is a cis face where it receives vesicles and a trans face where it sends the vesicles away. I'm gonna click and see if it'll show me. It does not, okay. So I'm pretty sure on the left-hand side where it looks like it's ready to catch something, that's gonna be the cis face, where it's receiving things, and the trans face, it goes out the other side. So the Golgi complex, this is kind of like FedEx. So it's going to get packages in from the endoplasmic reticulum. It may modify them, like put finishing touches, change some R groups, whatever. It modifies them, sorts them, adds some tags, and then packages them into new vesicles to exit via exocytosis. We know EXO means out, cyto means cell, so it's sending it out of the cell. Okay, so this modifying material step, this is so important because remember that form equals function. We've been hitting that hard this school year. So we have to make sure that the proteins are folded right before we send them off to do their job. So here is a Golgi apparatus. We see on the side that's curving in, we've got things coming in, and it looks like on the back side, there's some getting ready to be released. So that's kind of cool. Okay, lysosomes. So it's a sac with hydrolytic enzymes. So remember, hydrolysis breaks things apart. We added water to break things apart. So these enzymes are basically digesting things. So there's a concept called autophagy. So the cell can recycle its own worn out parts. So if there's, say, a mitochondria that it's worn out, so the lysosome can go and digest it, and those proteins and things that make it up can be recycled into another organelle. Remember, auto means self and phagy means eating. So it's basically eating and digesting itself. All right, so then there's a subset, like a category of lysosomes called peroxisomes. So these are super important, and only aerobic cells have them. But let's say that you got a little bit of peroxide in your body, or like you put peroxide on a cut and it bubbles. Those are your peroxisomes breaking down that hydrogen peroxide, that H2O2, and it makes a bubble. When it does that, it's releasing oxygen. So that's pretty dang cool. Okay, so here is what it looks like. So right here, we have a vesicle containing a damaged mitochondria and a peroxisome. So it is going to break down that worn out mitochondria. So it's damaged. It doesn't look quite as sharp as the other one. It's going to release those digestive enzymes and break it apart, which is pretty cool. So vacuoles, they're large, just really big vesicles. There can be food vacuoles. So if it eats like a big bacteria, that's in a vacuole until it can digest it. So contractile vacuoles help maintain water level. So if I need to add more water, or like the cell's starting to dry out, I can release water from that vacuole. So like it contracts and releases the water. Then a central vacuole is found in plants. It has inorganic ions and water, so like dissolved salts and things. These are the big water balloons that you see in plant cells. So like when you leave vegetables in the crisper too long and they get kind of limp, that's because it has lost water out of that central vacuole and lost something called turgor pressure, which is another problem for another day. We got turgor pressure right there. So turgor pressure, that's just the water pressure in a cell. I guess we'll go into it briefly. It's what helps keep the walls, the cell wall, rigid. So basically I need that pressure like from inflating a water balloon inside the cell. All right, so here is our electron microscope image of the large central vacuole. I just want you to kind of notice from all these cell pictures we've been looking at, cells aren't nearly as tidy and symmetrical in real life as they are in the diagrams. So that's kind of why I'm showing you these microscopic images, just so you can get a feel for what it actually looks like. So these other big organelles, those are its chloroplasts. They are super big, like they're not little green beans. In real life, they're huge. And probably the nucleus is that bit between, like in the middle right there, like right after the vacuole. So this word vacuole, like vacuous or vacuum, like it means empty, right? So it's like a big empty area when you look at it under a slide. Okay, so we are going to stop there. You're probably so tired of hearing me talk. So what I want to do real quick, let's bring up your, what's the word? Right, case study. See if this will bring it up. There we go. So because it is almost Halloween, we are going to be talking about a real life case study where something went wrong with an organelle, all right? This is a true story, like a whole bunch of people died when Tylenol was laced with cyanide. So we're going to be talking about this a little bit. It's kind of a precursor to what's going on tomorrow when we talk about energy organelles. So yeah, I want you guys to take a look at it. Just read through it and answer the questions as best you can. So good luck, and I will see you guys tomorrow with the other half of our cells.